Hi everybody, I'm back again, and today we're going to continue in our genetic traits unit, and we're going to continue looking at different ways that we move beyond what Mendel understands about genetics. In fact, today we're going to talk about learning goal number 10 and the ways that if you combine different genes, how it can change the normal 9 to 3 to 3 to 1 ratio that we would expect. So you should go ahead and turn to Big Ideas page 5 and work along with me. So, first of all, we can assume, or we can use the fork line method if you're trying to figure out the phenotype or the probability of a phenotype when there's more than one trait. If you know those two different genes are on different chromosomes and they undergo independent assortment. I want you to try to do this yourself with blood type and albinism. Take a second and use that fork line method to cross two humans that are both heterozygous for the autosomal recessive allele that causes albinism, and both of these individuals are also blood type AB. And try to figure out the phenotype ratio for me. When you have finished this, then we'll sort of discuss how discuss our results. But for right now, pause me. Okay, so I'm assuming because you're back, you've taken the time to cross two individuals that are heterozygous for albinism and they both have AB blood type. The results that I would expect that you got are something along the lines of this. So first of all, look at one trait at a time. Initially, look at albinism. You could have gone in the other order, and that would be correct too, but I started by looking at albinism. Each of these heterozygous parents have a 3/4 chance of having a pigmented child and a 1/4 chance of having an albino child. So you know that, and you break up the offspring into 3/4 pigmented and 1/4 albino. Then you take a look at blood type. And when you cross two AB parents, their offspring are going to be 1/4 type A, 2/4 type AB, and 1/4 type B. Again, these are predicted ratios, not exact. So we go ahead and plug those numbers in as the second part of the forked or branched diagram. And you do that twice, so that same ratio is going to appear in the pigmented group and in the albino group. Then you multiply going across, so you have 3/4 times 1/4, meaning we have a 3 out of 16th chance of having a pigmented type A child. And again, multiplying across, a 6 out of 16th chance of having a pigmented type AB child. And a 3 out of 16th chance of having a pigmented type B child. The same pattern continues, only now in the albino group of children, so 1 out of 16 chance of having an albino type A child, and a 2 out of 16th chance of having an albino type AB child, and a 1 out of 16th chance of having albino type B child. So here's our final phenotypic ratio. It's a 3 to 6, or a 3 to 6 to 3 to 1 to 2 to 1 ratio. Again, that's different from our normal 9 to 3 to 3 to 1 ratio. And that's because these are not both Mendelian traits. Albinism is a Mendelian trait, but blood type is not a standard Mendelian trait. And when you combine the two, you're going to get a different ratio. And that's what we're going to play around with today. So, moving forward, we're also going to talk about polygenic inheritance. When you have traits where you have a very clear phenotype, but it's influenced by more than one kind of gene, we're going to call that polygenic inheritance. And this is a result of gene interaction. Each of the cell products that come from each of these genes, the cellular function of the gene products for each of these genes work together to come up with one phenotype. So the classic example of this is human height. I want you to go ahead to the back of the room right now and line up according to height. And then have a conversation as a class that takes maybe 1 minute. What kind of pattern do you notice? Go ahead and do that, and then pause me when you're ready. Pause me until you're ready. And then turn me back on. Okay, I'm assuming that you have, at this point in time, lined up in the back of the room and you are standing from tallest to shortest. I'm guessing it looks like sort of a gradual line where there's people of a whole range of heights, probably ranging somewhere between, I don't know, just below 5 feet to somewhere just above 6 feet, based on past times I've done this with my classes. Now, if you move outside of this class, we're going to see people that are even taller than this, like Yao Ming. He's 7 feet 6 inches tall, and Danny DeVito, he's only 5 feet tall. And so we're seeing this broad range, and if we expanded even bigger than, say, famous people, then we're going to go and you're going to see Mr. Bao. He is 7 feet 11 inches tall, and Mr. Ping Ping, he's 2 feet and 5 inches tall. So again, in the human population, we have this huge range of possible phenotypes. So that is one indicator that this is a polygenic trait. I'm hoping as you look around the room, you're going to see that there are, again, the people that are taller and the people that are shorter. The people that are taller in the room have more genes or more alleles that code for being tall, and there's a collection of them. People who are a little bit shorter have less of those tall alleles and maybe more of the short alleles. And it is the combined effect of what they have that produces the phenotype that you see or their height. So again, polygenic traits have multiple genes, and it's the combination of the proteins produced by those genes that gives us our phenotype. We know something's polygenic when we see a huge range of phenotypes, not just 3 or 4, but instead of almost an infinite range of phenotypes, just like there's probably an infinite number of heights that humans could be. And other examples besides height are skin color, hair color, eye color, and we'll talk about another example in our lab. So let's take a look at a hypothetical example or a hypothetical model for how height might work in humans. So I want you to read about that hypothetical height model, and then I want you to try to do this. I want you to rank the following people in order from shortest to tallest based on their genotype. Go ahead and pause me until you're done. Okay, it looks like I may have gone from tallest to shortest, which could be confusing for you. But when I ranked them from tallest to shortest, I have AJ here. He has 6 of the tall alleles. Then I have Andre with 5 tall alleles. Notice in this hypothetical model there are 3 genes for height, and the A, B's, and C's are considered tall alleles, and the X, Y's, and Z's are considered short alleles. Andre had 5 tall alleles, Morris had 4, Charlie had 3, and Mateen had 2. And that's how we could rank them by height. I'm wondering if anybody guessed who these 5 individuals were and why Mrs. Polonsky might have picked them for this example. Well, if you have any Spartan basketball fans out there, they are the starting lineup for the MSU's, the year of 2000 National Championship basketball team, which was the year I graduated from MSU. And I got to be there when they won the national championship. And these guys, I actually ranked them by height. So you'll notice Mateen Cleaves right here. He is actually shorter than I am. And he made it into the NBA at least for a year. So let's try another example. Again, this is hypothetical. However, I did rank them appropriately by height. Let's see what happens. What genes would all of AJ's gametes get? Give that a shot, and then unpause me when you have an answer. Okay, hopefully you know that AJ in this hypothetical model has all tall alleles, which means that's all that he can give his offspring. He is going to give each one allele from each gene pair to his offspring. So no matter what, the only thing he has to give from this pair is an A. The only thing he has to give from this pair is a B, and the only allele that he has possible that he has to give from this pair is a C. Now, that was a simpler example. Let's go ahead and take a look at if we used Andre. Now, Andre, what are all the possible combination of Andre's gametes? Go ahead and pause me till you finish. Okay. Hopefully, you came up with 2 possible answers, because we know no matter what, Andre is going to give an A on this gene, because that's all he's got. And no matter what, he's going to give a C on this gene, because that's all he has. So this middle gene here is the one that gives us a little bit of choice in terms of our gametes. So we are either going to get an ABC gamete or an AYC gamete. This ABC, of course, has more tall alleles than the AYC gamete. And so we're going to go ahead and try a lab that has to do with the polygenic trait that you all have, and there'll be more details to come on that.